Oh, good morning, everybody. Just give me a second. Got to put my lights on. Otherwise, it's like I'm speaking from the bottom of a cave or something. Pardon me. All right. Boy, oh boy. Feels like it should be spring break already. But it's not. We have to wait all the way until May, which kind of sucks. Yes. Well, it'll be all the sweeter for when it gets here, right? And that's kind of the way I look at it. I'm going to be partying like it's 1987 or something like that. Okay, so let's see where we left off last Monday. I can't remember the day of the week, to be honest. Isn't that crazy? Okay. Oh, you don't need to see my email. You want to see this. Aha. All right. So, kind of to recap, right? We start off with, crap, I closed that one. Badgers. Okay. So essentially we start off with what's called a pre-mRNA. And so pre-mRNA is what is transcribed directly from a eukaryotic gene. Now remember, this is eukaryotes, right? This is not bacteria. Bacteria do things differently. And what we end up with, and this isn't my picture, by the way, it's far too good for that, is after all of that processing has occurred, we end up with what's called our mature mRNA. This is what is able to be translated. It isn't translated yet or able to be because it's in the wrong place. It has to be exported into the nucleus, out of the nucleus, sorry, into the cytoplasm before it can be translated. But it looks something like this, right? We have our 5' cap. Actually, let's ask the class, what is the 5' cap important for? A little bit of a recap. Two things. What's the 5' cap for? We covered this on Monday. Now, think. Also allows me to drink some of my coffee too. 5' cap, 5' MPG or methylguanosine cap. What is its purpose? Two reasons. Anybody remember? All right, so what happens to bare mRNA that has nothing on either end in terms of caps or proteins or whatever? What enzymes are around which will recognize it and like it a great deal? Kind of. There'll be enzymes that will degrade it. So these are called RNases. And most of them recognize the ends of mRNA, right? So if those ends are bare, that RNA will be degraded, right? And so we'll get to this a little later in the course. RNA stability is actually a key part of the control of gene expression, right? This is why I keep going back to it, because it's kind of really important for that purpose too. What about translation? So this mRNA has to be translated at some point. What does the eukaryotic ribosome recognize? Which part of the mRNA? If I'm asking about the 5' cap, I'm kind of telling you the answer already. Oh, it's the 5' cap, right? So 5' cap is important for stability and for ribosome binding later on in translation. 3' end, we have a poly A tail. Haven't got to that yet, but we will do this class. And in between, we have two UTRs. So I might have written this up before. I can't remember. And it simply means untranslated region, right? So this is also important for translation and mRNA stability, depending on which end you're talking about. And then we have our ORF in the middle, which is our open reading frame. Essentially, that's the bit that gets translated. Can't see what I'm doing off. Need a bigger screen. And CDS essentially is the same thing. That's a coding sequence. Right, so this is what we end up with. It's not what we start with, right? So the very first thing that happens, I could do that. Neat. Shrink the chat box a little bit. Oh, and also we have our start codon here, which is an AUG, and we have our stop codons here, which are one of these three different combinations of bases. Right, so as soon as the transcript kind of appears, right, once it's made its way out of the RNA polymerase, we have the capping machinery recruited, and that protects it straight away, because otherwise it's going to get degraded pretty much as soon as it's produced, which will be obviously a waste. And then as soon as an intron appears, we have splicing machinery start to assemble. Okay, and so this is kind of where we left off. And there's a bunch of stuff involved. The whole thing is called a spliceosome, right? And those are made up of a bunch of one of my favorite acronyms, which is SNRPs. Kind of pronounced the same way as schnapps, if you've ever had that. It's like a really nice German liqueur made from almonds, I think. I can't remember. A long time since I've had any. Anyway, so these SNRPs are made up of RNA, so ribosomes essentially, right? So that's these small nuclear RNAs, which are produced by RNA polymerase II, same as mRNAs. And we have 5 of them that are involved, right? So these 5 RNAs make up 5 different SNRPs, and they recognize different parts of the pre-mRNA to create, go through and do that splicing job. Okay, so one of the reasons why we talk about splicing is because A, it's really important. And if you get it wrong, that gene simply, it produces a protein that doesn't work, right? So getting this right is really, really important. And also, there are multiple kind of introns and exons in most eukaryotic genes. So that means that you can actually shuffle and mix the order of exons to create different proteins from the same gene, which is pretty cool, right? And that allows you to do essentially different things with the same set of instructions. Okay, so we're going to go through the mechanics of splicing right now. And this is fairly involved, right? And really there's some key principles involved. One of the main ones is what are called these splice acceptor sites. So there will be one at the 5' end of an intron and at the 3' end of an intron. Right, so this is essentially the first intron in a gene because we have exon 1 here and we have exon 2 beginning over here. And these sequences are highly, highly conserved, right? And essentially it's in DNA it would be a GTAG. So there's a GU in the RNA here and an AG here. And so CAG actually is the full version. And these are what are recognized by the spliceosome, right? In a kind of similar way to the way transposase recognizes the inverted terminal repeats on a bunch of transposons, right? It's kind of same sort of idea, essentially. And the funny thing is when we mean, whoa, where'd that go? I didn't realize I was holding on to that. When we talk about something that is highly conserved, right, what we actually mean is that any changes to these bases, particularly the GU and the CAG, render that protein or that gene non-functional. And so if that protein or that gene and therefore that protein are involved in something important, that mutation never spreads through the population, right? And so all of the gumph inside here, other than the letters that have been highlighted, has no selective pressure on it whatsoever. It could be anything, right? Just complete gibberish, right? But these few bases are absolutely required for this process to work. So any mutations that change these sequences stop the splicing from happening. They're actually called splice acceptor site mutations, right? And so they're so conserved because essentially natural selection has over millennia selected very strongly against any mutations that change these sequences. Right, and so then in a sense, when we talk about consensus sequence, we're also talking about how important these bases are for that particular function. And we're also making a kind of a statement about evolution as well, in a sense. So these two ends are super important, the GU, CAG, and there's also a few others in here which are important as well. And the third thing that's important is this A, this adenine here, which is a short distance upstream from the 3' end of the intron. This is really, really important, and this actually ties into the whole funkiness of RNA. Okay, so here's another question for class. Just make sure you're all still awake. What are some of the differences between DNA and RNA? Two key differences. Have a think. What are the differences between DNA? That's right. Cool, Jimena, that's the first one. So in RNA, you have a uracil. What's the other big difference? Down to the structure of the sugar, or really what's attached to it. And the reason why DNA is deoxy and RNA is not. Yeah, DNA is double-stranded, but RNA can also be double-stranded too. RNA can form all kinds of funky structures. So that's kind of a ooh, sort of, yeah, kind of maybe. It's the other one. The one that is absolutely cut and dried, definite difference. Think about the 3' OH in DNA and RNA, right? That's shared. Is there another OH which is not, which is present in one and not present in the other?
Present in DNA.
It's not present in DNA. That's the deoxy part. So yeah, the ribose, Jimena, is what these OHs are attached to. And so in DNA and RNA, the 3' OH is involved in nucleotide extension, essentially kind of synthesizing that strand of nucleic acids. The 2' OH is only present in RNA. That's okay. So deoxy means basically taking that oxygen out, which is why DNA is called DNA essentially. So the 2' OH is present only in RNA. Oops. Right, which for one thing makes it more fragile, right? Because it's a site that enzymes can attach and kind of add things to. But in this case, it's really important because we're actually going to be kind of attaching the end of this intron to itself. And so this is blown up here. So this is the 5' end of the intron here. And you can see the phosphate group on that, attached to that 5' end of that sugar, is now going to be attached to the 2' carbon of this adenine. Right, and so you can't do that in DNA because there is no OH group present on that 2' carbon with which you can actually add this thing to. Right, and so this is a kind of a key fundamental difference between RNA and DNA, right? And it's kind of a neat example of showing why that matters in this case. So the very first step of splicing is to cut the, well, first you have to recognize those splice sites, obviously, and we'll go through in a little bit more detail in a second. But you recognize that splice site and you cut it, right? So you physically break that pre-mRNA strand apart. Oh, I should put my hand like that. Then you curl the 5' end of that intron round and stick it back to itself at that adenine, which you can see just here. Then the next step is to cut this 3' end of the intron, which gives you what's called a lariat loop, which looks like a lasso. I always like talking about that in Texas. And then you stick this exon 1 to the end of exon 2. This intron is then degraded back into its constituent nucleotides, so those can be used again. So it's a multi-step process. As you'll see in a second, there's multiple different things involved in doing it. But the general principle is you have to take this out, but you can't do it in just one go. You can't just simply cut out the whole intron and stick exon 1 to exon 2. You have to first cut one end and then the other end and stick those two together, because essentially you don't want those kind of ends just flowing off into the nucleus or the cytoplasm. You need them to kind of stay in fairly close proximity to each other. Does anybody have any questions so far? Questions. Okay. So the thing that does all of this, or the things, are SNRPs. Right, and SNRPs are small nuclear ribonuclear proteins, which is why I call them SNRPs. Right, their first job is to recognize, and this kind of makes sense, right, because these are the first things that happen. They need to recognize the 5' splice acceptor site here and the adenine at the branch point. Right, so that's done by U1 and U2, right, which you can see here. And essentially what then happens is we have the other 3 SNRPs come in, and in a way that I don't actually fully understand, just going to make a note, look up something. Essentially what happens is U1 cuts the intron at the 5' site. Then with the help of the others, or these SNRPs coming in, it bends that mRNA round and sticks it to the A point, but at the same time, right, and this is the key, one of the key points, exon 1 stays attached to the spliceosome. So exon 1 is never allowed to go off on and have its own adventures. It's very closely tied to U6 and U5. Right, so once that's been done, I'm pretty sure it's U5 that does the cutting and religating, but I'm not 100% sure about that. Essentially, the 3' end of the intron is released. And because exon 1 is still kept in close proximity, the end of that exon 1 is now able to be religated, basically formation of another phosphodiester bond to the start of exon 2. And then all this other jazz gets released. The intron gets degraded, and these schnurps are then free to go and assemble and process another intron, right, that's being excised. So again, a stepwise process, cutting the 5' end, forming the lariat loop, cutting the 3' end, and then sticking exon 1 to exon 2. So you can see how carefully orchestrated this process is. Okay. So essentially what we have is a series of events, right? These don't necessarily have to happen in a particular order other than the capping of the 5' end, right? That has to happen first. And essentially, as RNA polymerase trundles along, kind of making the pre-mRNA transcripts, right? And essentially here you can see these darker patches correspond to exons, the lighter patches to introns. And so as RNA polymerase goes along, these introns appear, and depending on what's around at the time, these introns will get spliced, not necessarily in the order in which they're produced. Also, another interesting point is that these splice junctions, right, between neighboring exons also serve as a binding point for the proteins that are involved in RNA export. So they serve kind of two purposes, right? Not just getting rid of the introns, but also facilitating export from the nucleus. And then the very last step is the addition of what's called a poly-A tail, right? Which happens after the transcript has been released by the endonuclease. Or if you remember back to termination of transcription, RAT1, that mRNA transcript is pre-mRNA, transcript is cut and released even while RNA polymerase is still synthesizing RNA. And once it's cut and released, now we can add our poly-A tail. And this is another important feature of a mature RNA, because this serves as the binding site for what are called poly-A binding proteins. And those protect the 3' end of the transcript from degradation. So essentially, what you have is kind of transcription, right? That pre-mRNA is cleaved, right? And that's done via essentially a consensus sequence near the end of what will be the mature transcript, and a U-rich sequence. I'm not entirely sure what the purpose of that is. I don't know if that's to slow things down or what, but anyway. So the pre-mRNA is cleaved here, and then you have a process called polyadenylation, right, which is simply the addition of lots of A's. And essentially, you have I'm not even sure what does that, to be honest. Polyadenylase would be my guess, but it must be some kind of A-specific RNA polymerase that just adds a stretch, sometimes a few hundred bases, right, of adenines to the 3' end of the transcript. And this can be, they can be from 10, 20 to several hundred base pairs long, right? And again, this serves kind of in the way that telomeres do, sort of, not entirely analogous, but it serves as like a buffer at the end of the transcript, right? And those poly-A binding proteins protect it from RNases. Okay. Cool. And so actually one of the kind of really interesting kind of dials by which gene expression is controlled is controlling the stability of the mRNA once it's produced, the mature RNA. And there are lots of different ways that you can do that, but one of them is modifying how well those PB poly-A binding proteins bind to that tail. And so if you want to get rid of a transcript, kicking them off is a very effective way to get that transcript degraded so gene expression stops. Okay, so again, this is kind of a word description of this, which we've already covered, and now you can see how we get to this particular endpoint, where we have our caps, we have our poly-A tail, we have no introns present, which is super important, and we have our UTRs, which are really important in translation and RNA stability. Okay, where are we at in the schedule? I think we're a little ahead of time. What's today? 31st. Yeah, we're way ahead of time. We'll have plenty of time for recap later. Anybody got any questions about any of this or stuff that we have covered up to date? Could be about any single thing you want to ask about. Because now's a good time to ask, right? What a great day. Okay, so now we need to get on to translation, right? And translation is a big confusing topic. I mean, in many ways, translation is pretty simple, right? You take your mRNA and you end up and you use that as a instruction set for assembling a sequence of amino acids, which then kind of, fold up in their whatever way and produce a mature functional protein. Right. So in concept, it's really pretty straightforward. In practice, there's a lot of moving parts that do very specific things, and there are some really key differences between prokaryotes and eukaryotes. Ooh. Mainly, sorry, I'm kind of a little bit wiped out today. Mainly in the initiation of translation and, a few other things too. And so essentially, our building blocks for translation are amino acids, right? And this is something that we covered, or you would have covered in GenBio 1, talking about macromolecules. And essentially, amino acids consist of what's called the backbone, right? Which is an amine, a carbon called the alpha carbon, and a carboxyl group. And this is the same for all amino acids, right? Even though we have 21 different amino acids, it's the same for all of them. And it's actually very much analogous to DNA, where we have a phosphate sugar, which is the same to all of them and forms the backbone of DNA and RNA, and the nitrogenous base, which is the thing that's different, right? So the analogy is pretty good between those two different kind of units of macromolecules, right? And so this is why it's called an amino acid, because we have an amine group at the end, or the amino group, and we have a acid or carboxyl group at the other end. This also is what determines the directionality of proteins. And so proteins have direction, and we have an N terminus, start, and C terminus, end of a protein. And so the N terminus is obviously from the N in amino, or the actual nitrogen. And the C terminus is the carboxyl end, right? And so new amino acids can only be added to the C terminus. For very much the same reason that new nucleotides can only get added to the 3' end because of this hydroxyl group here. Right, so this is what's used to create that peptide bond. So, okay, here's another question for class. What's the name of the bond that joins nucleic acids to each other, like one nucleotide to another? What's the name of that bond? It's not James. It's not Roger Moore. Because they're all bonds, right? You probably don't remember Roger Moore. What's the other one? Daniel Craig. Although he's looking a little bit craggy now that he's getting older. Not that I'm jealous or anything. Yeah, thank you, Sophia. It's a phosphodiester bond. Thankfully, you stopped me from going too much further in my terrible bond jokes. Right, so the peptide bond is the same as, well, what works for amino acids and peptides as DNA and RNA. So, oops, peptides, PEP. Join amino acids together into essentially a polypeptide chain. And so obviously, you know, when you start, you have 1, then 2, and then 3, and then 4, and so on. You can have potentially thousands, right? So some proteins are huge, consist of thousands of amino acids. And some proteins are even huger, if that's a word, bigger, there you go, and have multiple huge proteins kind of just sticking together. Actually, ribosomes are one of the largest proteins that, or protein assemblies that you'll see in the cell. Okay, so kind of, and also before I forget, this side chain R, I'm half tempted to tell you my pirate joke, but maybe you've had enough jokes already. This is what's different between the amino acids, and this is really what gives amino acids their properties, whether they are hydrophobic. Do we have? Oh yeah, we do. Here we go. Whether they're hydrophobic, right? So if we kind of blow this up a little bit. So these would be side chains that are not able to form bonds with water, right? So we've got lots of carbons and hydrogens and very few oxygens and nitrogens, right? And so where in the protein, right, given that proteins are kind of globular, right, make balls in some sense, and our cytosol, our cytoplasm is largely an aqueous environment, where would you expect to find most hydrophobic amino acid residues in the protein? If you think about proteins in a 3-dimensional shape, where would you expect to find these amino acids? Are they going to be on the surface of the protein, or are they going to be on the inside of the protein? The opposite generally. So our cytoplasm, that's all right. Our cytoplasm is aqueous, right? There's a lot of water in there, right? That's, you know, what makes our cells nice and plump. And also why, you know, if you want to preserve things, you desiccate them, right? You stick them out in the sunshine until they're nice and dry. But inside our proteins, you'll tend to find hydrophobic residues because you actively want to exclude water, or A, they're there because they don't want to be in contact with water, right? And they would just cause problems if they were. But also, you want to exclude water out from the center of proteins. It can be very disruptive, right? And actually, one of the things that causes proteins to denature, like deep down in the sea, there's probably a song from The Little Mermaid there that I could sing. Probably sure you don't want me to either. But basically, one of the main problems with Under the Sea, that's pretty much all I can remember actually of it. It's a neat song, but I can't remember anymore. One of the problems with if we take one of you and we stick you with, you know, face mask or something at, I don't know, 1,000 meters depth, is that other than the fact that you will get squished, right, by the pressure, and it'll be extraordinarily hard for you to breathe. The other issue is that your proteins, all the pressure is going to be forcing water into the center of your proteins, and it just forces those proteins apart, right? And so under normal conditions, these hydrophobic residues help prevent that, right? They keep that core of the protein nice and water free. But you also have hydrophilic side chains, right? So these have OHs and NHs, right? You can see them over here. And these are able to form bonds with water. So you tend to find those on the outside. Or if you have a channel, right, which allows water to pass, that channel, right, which they're often like actually like barrels, the core of that channel will be hydrophilic, right? Because then that will allow water to pass through it into the cell or out of, depending on osmotic pressure. And also we have charged side chains, right, which form ionic bonds. Actually, where the hell's cysteine? Now I'm thinking about it. Going back up you get. Where do they put cysteine? Oh, they put it there. That's kind of weird. Well, cysteines actually form disulfide bonds, right? So the sulfur here is able to form a covalent bond with a matching cysteine somewhere else in the protein. And so those are extraordinarily strong bonds. They're as strong as the bonds between amino acids. And you tend to find those wherever you need like real rigidity and structure in a protein. And then you have these which are able to form ionic bonds. Quite often you tend to find these pairing up with metal ions, right? And so an awful lot of proteins that do cool things in terms of reactions have metal ions in their cores. So one of those ones which you're probably most familiar with is hemoglobin, right? And so hemoglobin has an ion, an Fe ion at the center of that protein, and that's what allows oxygen to bind to it and get released from it too. Other ones are there's all kinds of iron and sulfur and copper clusters in your electron transport chain, which are involved in passing electrons down the electron transport chain to generate a proton gradient and then therefore ATP, right? So which amino acids you have where plays a huge role in both the shape and the function of that protein, right? And so this is why some mutations are not super critical, right? So, for example, other than obviously the silent mutations which don't change an amino acid, if you went from glycine to alanine, maybe nothing will actually change in terms of function, right? Because you go from having a very small to a pretty small side chain. right? If you went from glycine to tryptophan, which is basically small to huge, you may disrupt things. If you go from hydrophobic to hydrophilic, or even probably worse, um, a basic or acidic side chain, you'll almost certainly disrupt the structure and therefore the function of that protein. Right, so that's why, depending on where those mutations occur and what consequence they have in terms of amino acid sequence, those mutations can vary from being hugely important to being, nah, whatever, kind of damn squib. Pardon me. Okay, so let's kind of turn that back a little bit smaller. Here we go. So essentially, there's a bunch of players in translation, a bunch of things which are critical for it to work. The obvious one is messenger RNA, obviously, because that contains the information needed to create the correct order of amino acids in that polypeptide. Right, and so you're probably wondering, why do we talk about polypeptides and proteins, right? What's the difference essentially between them? And so polypeptide, and that is essentially the sequence of amino acid residues in a protein. And so this is what is produced by the ribosomum. There we go. Now, protein is a three-dimensional structure, and they vary very, 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 very much in terms of what they look like. If I can spell structure. That is the result of a complex folding process. So essentially, the polypeptides are the kind of the beads of a necklace, right? That's the actual order of those amino acids. A protein is when that kind of string has been kind of bent up and folded together to create a very specific shape, which does a very specific thing. Now, protein shapes can vary hugely, right? So you've got, I don't know. No, not videos. Don't want to watch video. We've got myosin, right? This is one of the proteins that's involved in muscle contraction, right? So this is what myosin looks like, kind of down here. This is what actin looks like, right? So again, two very different proteins. This is a long kind of twisted helix. This is clumps of fibers with little things on the end. Just trying to think. This is a protein involved in glycolysis. Yeah, glycolysis. I think it's in glycolysis at some point in there. Right. So now we get a very different structure. Right. And this is a kind of sheet and ribbon model, rather. There's NAD. And you can see, okay, we've got a very, very different structure now. And then we have, oops, one of my favorite. This is a super cool protein. This is actually consists of 4 different proteins, alpha and beta chains, and each one of these has an iron, Fe, iron in the middle of it. Right, and so these actually change shape depending on whether or not oxygen is bound or not. Right, so there's a huge difference between one protein and another, right, in terms of its structure and shape. And so all of that information is coded by the mRNA, but it's the amino acids and the order in which they're in that create that structure. Now, the other things involved in translation are the enzymes and structures and RNAs that are involved in the actual translation process, right? So ribosomes you know of already. These are a mix of RNA and protein, and it's actually the RNA that does the work in a sense. So ribosomal RNA is a ribozyme, and a ribozyme is an RNA that has catalytic properties. In this case, oh, what was going on with my ends? Sorry, spelling is not working very well today. And so the ribosomal RNA, or the rRNA, is what is actually responsible for doing the job of translation. The protein is there more to create the structure in which it can do that. tRNAs are our Rosetta Stone. So these are the intermediaries that allow the translation, in a very literal sense, of the information in mRNA into a amino acid sequence, into a polypeptide. Right, and so they do that by essentially carrying an amino acid, a very specific amino acid, to the ribosome, but also containing within them a little patch of 3 nucleotides called an anticodon that recognize individual codons in the mRNA, right, which are groups of 3 nucleotides. And also we have another enzyme, which is, these are amino acid tRNA synthetases. And so essentially what they do is they recharge tRNAs. So tRNAs go into the ribosome, drop off their amino acid, and then get kicked out. And so when they get kicked out, they need another amino acid, and they get recharged using these amino acid tRNA synthetases, which do a very, very specific job of adding only the correct amino acid to the correct tRNA. And essentially, it's another, oh, they're only 20. I always thought there was 21 for some reason. Yeah, that's polypeptides. Okay, so, and essentially what that looks like once you start kind of building it up is a series of amino acids, peptide bonds shown here in red, in a long linear chain, right? So again, this is as it's being produced. It's not necessarily what it will look like once that protein is fully processed. And here you can see the different R groups in that polypeptide chain. And that's pretty much it for now. We're a little bit ahead of time. Well, there's a funky picture. There you go. There's another picture of a protein, but we'll get to that another time. Does anybody have any questions before we finish up for the day? One thing that I'd strongly recommend is watching videos of this process, right? Because that will make it potentially quite a bit clearer what's going on. Once we get into the mechanism, it can get a little bit confusing, potentially. Cool. Well, all right. Thanks very much, everybody. I shall see you either in a little bit if you're in lab or on Friday. Have a great day.